afternoon to everyone, and thank you all for coming. I'm Nisa, and I'm currently a third-year PhD student under Professor Desvanto. So today I'd like to share with you my PhD project titled Liposomes as a Delivery Platform of Vaccines Against Group A Streptoccocus. So Group A Streptoccocus of gas, as you have heard from my friend Nirmal, it colonizes the throat and the skin. It's easily transmitted via small water droplets in the air, which infects millions of healthy individuals each year. It is responsible for a range of diseases, both trivial ones and lethal ones, such as the common sore throat, pharyngitis, such as also necrotizing fasciitis, which is a flesh-eating bacteria, and also rheumatic heart disease, whereby Australia's indigenous people suffers the highest rate of. Approximately 60 to 80 million people currently suffers from rheumatic heart disease, and it causes about 1.4 million deaths per year, which makes it one of the top 10 pathogens in the world today. So one of the major virulence factors of gas is the M protein, as you've heard. So the M protein is a major surface-associated protein expressed by gas, and immunity to this M protein has been proven to be protective against gas. However, we cannot use the M protein because it is also the region that is responsible for autoimmunity. Therefore, a P145 peptide sequence, seen here, was derived. However, we can't use that because it has similar sequences to the human cardiac myosin. Now, let me draw your attention to this schematic figure right here. This is the schematic figure of the M protein. As you can see, there are 4 different domains, A to D, and it is highly hypervariable at the N terminal. However, it gets less so towards the cell wall, the anchors here. So what we did was to get a minimal B cell epitope within the C repeat regions of the M protein. This epitope, highlighted in blue, this one, it loses its alpha helical conformation. Therefore, we added the GCN4 alpha helical promoting sequence to get the alpha helical conformation, and this we call J14. And this J14 is found to elicit protective antibodies against P145 without any cross-reactivity against the human tissues. So, as we know that gas is easily contracted via inhalation, what we wanted to do is to prevent colonization. In order of doing so, we want IgA responses. Therefore, one way in getting IgA antibodies is immunizing via intranasal deliveries. Other advantages includes its ease of administration, and also it elicits local and systemic antigen-specific IgA and IgG immune responses. Next, we know that liposomes itself enhances salivary IgA. Therefore, we use this as one of our delivery vehicles. So it has other advantages, such as its ability to protect its active ingredients from degradation. It also has the ability to incorporate both hydrophilic and hydrophobic antigens as well. However, it has weak adjuvanting properties, and we know that peptide alone is not immunogenic enough. Therefore, we also use the lipid core peptide, and this lipid core peptide is made of the lipid core polylysine dendrimer, whereby we could attach our multiple epitopes right here. Other advantages of this lipid core peptide is its ability to activate dendritic cells. It also improves stability towards enzymatic degradation. It is easily synthesized chemically and on a larger scale. It is also very stable under freeze-drying conditions. So with that, I'll explain my vaccine candidates. I haven't mentioned P25. P25 is a universal T helper epitope. So in my liposomes, I've incorporated J14 and P25 in one of the constructs. The next one is a conjugate compound, which I call lipopeptide 1. It has 2 C16s and conjugated P25 and J14. And then I would like to compare it with the peptides separated with only 1 C16 and a double C16 here. So once I've finished synthesizing the antigens, I've incorporated them into liposomes. How I formulated liposomes is slightly different from my colleague Nirmal. First, I put the lipid component in chloroform, and I got rid of the chloroform, and I freeze-dried it, and then I hydrated it with the vaccine antigen. And from there, I regulated the size using the extruder. I used a 100 nanometer pore membrane, And this was my results for size. And then I tested for zeta potential as well. All the formulations had approximately 60 MV, and the size was about 140 to 160 nanometers in size. So this is a TEM of liposomes LP1. That's my major vaccine candidate right there. What I've noticed is that vaccine candidates that had lipids had a higher loading efficacy. This was done by ultracentrifuging my liposomal vaccine candidates, and then I would lyse the liposomes and calculate the concentration using by a standard curve. So here's my immunization schedule. It's pretty similar to my friends. However, what I did was that I extended it for 5 months, and then I did a re-stimulation, and I've only boosted the mice twice. So I've collected serum after 14 days after every immunization and saliva 1 week after every boost. So I've used Swiss mice because we have the P25 epitope, and each group of animals contained 5 mice, and we've loaded about 30 micrograms of peptide in 15 microliters of liposomal solution. So here are my results. After 3 immunizations, let's just focus on the LP1 right now. It is actually significantly higher than my positive control, which actually had CTB inside. So that was really, really good results for me. However, other things that I'd like to point out is that all my liposomal formulations were significantly higher than the PBS as well. So it shows that there is some sort of improved delivery, at least. Next would be the measuring IgA titers. Once again, I've got really high titers for liposomes with LP1, the conjugate, and it is significantly higher than PBS, which is here. However, the other liposomal formulations, some of them did not show any significance to the PBS. What I wanted to know was where was this direction leading to, so we did ELISAs for IgG subtypes. And what we did was to test IgG1 and IgG2A. What I found out from literature is that the ideal response in a vaccine is that we want to balance Th1 and Th2, because Th1 drives the inflammatory responses, whereas Th2 is the anti-inflammatory. So what we wanted, ideally, is a very balanced response, and which was what we got. Right here, liposomes LP1. Without the liposomes itself, we got very low levels of IgG2A, and we know that we wanted the pro-inflammatory cytokines for opsonization of our bacteria as well, which we have already published previously that LP1 itself was able to opsonize gas bacteria. We then moved on to a long-term memory study because you want a vaccine to last, to be able to produce antibodies on a long-term basis. So what we did was, after 5 months, I collected blood, and then after that, I re-stimulated mice that were already primed with liposomes LP1 and the PBS mice with liposomes LP1 itself. What I've noticed is that with just 1 immunization, I got really high IgG titers for the PBS mice, and these were already mice that were really old. So, however, I couldn't see any significant difference between before re-stimulation and after for the IgG results. However, for IgA responses, we had significant differences when we actually re-stimulated the mice with liposomes LP1, which is really, really great because it suggests like there is some sort of memory responses here. However, we did not get much IgA titers with 1 immunization. So in conclusion, I've successfully synthesized and purified all my peptides. I've also did an encapsulation efficacy test, and the encapsulation efficacy increased with antigens which had lipids on them, and mice immunized with liposomes LP1 had the highest IgG and IgA responses in comparison to all of my other formulations. I did manage to get a balanced Th1 and Th2 response. There were persistent IgG titers. There was significant increase in salivary IgA in mice immunized with liposome LP1, which I've mentioned suggested memory response. And combination of LCP and liposomal delivery platform has shown excellent efficacy in inducing long-term mucosal immunity and memory responses against group A strep. So I'd like to thank everyone involved in this, especially Professor Isvan for giving me the opportunity to work in his lab, Dr. Mariusz Kwaczynski, who is my co-supervisor, my other lab members, UQI for the scholarship, also people at Griffith University where we did our animal studies, Professor Michael Good, Michael Betzloff, and all the animal house staff members. Thank you.
So, two questions. The first is, did you check all your preparations for endotoxin? Because obviously that could be explaining the TH1 skewing of the response.
I did not check for endotoxin.
So I think that's important to go back and, because that's a very common laboratory area in these studies. The second thing is these antibodies, I presume, are bactericidal that you're inducing. So in your ELISA, you measured, I presume, the responses to a peptide.
Yeah.
But you need to be sure that those antibodies are actually able to bind the bacteria and ideally do a bactericidal assay. So
yeah, because previously, just with the LP1, it was done already, and yeah, we didn't manage to do the bacterial for liposomal constructs, but we have the ones for the conjugate itself without the liposomes.
But yeah, each time you change the construct, you may denature the epitope or change the epitope, and so it's absolutely essential that you show that that those titers are true when you actually look at binding to the bacteria, not just to a peptide.
Thank you.
Do we have any other questions? Your volume that you used in the immunisation, was that 15 microlitres, you said?
Yeah.
So what do you think the actual route of vaccination was then?
That I'm pretty sure it's overloaded and it went to the gut.
Because anything more than 5 microlitres per knee is going to go into the lung and also the gut, so you've got a mixed...
I thought about it, and I did check the fecal IgA if there was any, but there wasn't, which was a bit, I don't know, because previously they did report there was. But this time around with my liposomes, what I think is that because I've used cationic liposomes, hopefully it actually interacted with the membranes more, and that it took a longer time circulating inside instead of going straight to the gut and out.
Shirani?
Do you know how these things get taken up?
I did a preliminary DC uptake study, so that's what Nirmal presented. No, we didn't manage to do whether it's right at the nasal cavity or the lung. That's something we should look into, which we haven't.
How long was your thesis?
We actually just took spleens and just incubated them, and it was after a day's worth of incubation with our compounds. Most likely, because our particles are pretty small, it's less than 200 nanometers, which is needed to travel to the lymph nodes. So I'm guessing, yeah.
Any other questions? Okay, thank you very much.
Thank you.